So I'll just do a very brief intro. Welcome to the 8th annual Wheeler Lecture. This is the main event of the year for the department, and it's named after David Wheeler, who we remember as an eminent and pioneering computer scientist, but also as a colleague and a teacher and a friend to many in the department. We're delighted that Joyce Wheeler is able to be with us today, as she has been for many of these events. So our distinguished lecturer this year is Jane Hillston. Jane is Professor of Quantitative Modelling and also Head of School at the School of Informatics in the University of Edinburgh. She has far too many achievements for me to go through because it would take up too much of the time. But I want to just highlight one of them, which is that she was the recipient of the first ever Roger Needham Award in 2004. So today's talk is on combining formal methods and machine learning, and I'll hand over to Jane now. Thank you very much.
So thank you very much for the introduction and for the invitation. It's a real honour to be here to give the David Wheeler Lecture and to meet Joyce. So I'm a modeller. I model things. I want to understand the dynamics of how systems behave. And for most of my career, I've used formal methods. So I build models very much describing the behaviour of systems. But recently, when I go to perhaps social events and say I do computational modelling or I try and understand systems, people assume I'm probably doing deep learning and things. So this talk is trying to explain how recently, through collaboration with a machine learner in the department, and we've shared 2 PhD students, we've been exploring that interface and trying to understand it a little better. So I'm going to talk about 2 strands of work. One is putting inference in a more formal context, and the other is taking the machine learning statistical abstraction back into formal models. So what is a model? When we model things, essentially we're building some kind of function that takes inputs to outputs. We want to know if the system is parameterized like this, what do we see in the behavior? And in theoretical computer science, we tend to do that by building a real representation, and we have many mechanisms to do that, where we use discrete states and describe how the system behaves, sort of step by step. And our inputs there might be the parameters that we use within that model, and the output is the observations that we see. Whereas in machine learning, it's a much more data-centric approach, where typically we might take lots of examples of inputs and outputs, so the input-output relation, and then we'll try and impute the function that tells us how inputs turn into outputs. And these are often seen as really opposing ways of doing things. So what do I mean by model? Here's a very simple example that I'll use in the first half of the talk as a running example. So this is a model of gossip spreading. Okay, so we have some people who are, I like to say, innocent. My PhD student tends to call them ignorant. But we have some people who don't know the latest gossip. But if they meet someone who is a spreader, who has the gossip, they can interact through a spread action, and the innocent person will become a spreader as well. But gossip's only good when it's fresh. And so when a spreader meets other spreaders, they realize, oh, it's a bit stale, and they'll become a repressor. Or you may have some people who really say, oh, you should not be repeating that. And so we can have interactions between repressors and spreaders, where again, the spreader will become a repressor. Okay, so we have 3 different states for people in this system: innocent, spreader, or repressor, and we have these different actions that can take place. We can have a spreader interacting so that an innocent becomes a spreader, or we can have stop 1 where we interact with a spreader and become a repressor, or stop 2 where the spreader interacts with a repressor and becomes another repressor. And the models that I'm interested in, we will typically enhance them with timing and probability information, because what I want to know is if I start out with 1 person who knows the gossip and 100 people who don't, how long is it going to be until there's a 90% chance that most people know the gossip? So that's the kind of information that I'm interested in models. And so if I'm going to do that, I need to assign rates to the actions like spreading and stopping. And the usual process that we do for this is we'll build our state representation there. We may have some information, some data about how the system works, and we will run the model, see how close we are to the observations, try a different parameterization, and then do that until we feel the model is a good match. We've got a good fitting. And this is very much how disease modeling is often done. And then the opposite is the machine learning view, where things are much more data-driven. So we take a lot of data and we perhaps label that of, okay, this is a spread, this is a repressor, and we build the model in that way. I'm not particularly interested in deep learning. I'm much more interested in Bayesian learning because, as I said, I'm interested in processes with dynamics and probabilities. And so with the Bayesian learning, what we tend to start with is what we call a prior, which will be some kind of belief or hypothesis about how the system might be working. I then combine that with the data or observations of the system, and through inference reach a posterior, which is usually a firmer idea about how the system behaves. Okay, so we have Bayes' theorem, as we see here, which tells us that this posterior of updated belief based on the data is proportional to what we believed in the first place, and then the likelihood that this is the data given the parameterization of our model. Okay, so what I'm going to do is, as I said, talk about two different aspects of trying to bring these things together. And the first is where we essentially take the way that we do things in theoretical computer science, and particularly formal methods, and try and bring some of that into the inference world. And then the second is where we use some of the ability here to impute a function and use it to help us build a simpler model which is easier and more efficient to compute with to understand the behavior of a system. So there are many different ways of building these state representations using various different formal techniques, but I'm going to talk about process algebras because that's what I've done for most of my career. And the basic idea of a process algebra is that you have some agents who undertake actions, and we'll typically call actions lowercase letters and agents with big capital letters. So here we've got an agent who first does an action alpha and then behaves as the agent P. And what we have for that is a structured operational semantics, and I'm not going to go into details of the semantics today, but essentially we have a set of rules that tell us how to interpret such language descriptions and turn it into a state description. So we give it a labeled transition system, we say what states we have, what transitions, what actions move us between those states, and that gives us our discrete state representation of the behavior. So classic process algebras such as CCS and CSP focus on the possible actions without any notion of timing or probability. But I'm particularly interested in stochastic process algebras where we enhance our actions to also have a notion of rate. So we have some idea of how long and how probable actions are. Now this could get very complicated, but we choose to make our rates be the parameters of exponential distribution. So in other words, we say that durations are random variables that are exponentially distributed. And that makes our life very simple because it means that when we build our labeled transition system, when we look at the states and the way we move between them, it means we fall into a particular category of mathematical process called a continuous time Markov chain, which has a property of being memoryless. And so we don't need to keep clocks or anything. We can think of this as just like a state machine with some extra information and some extra ways of reasoning about it. So we build this continuous time Markov chain, which I'll explain in a bit more detail in a moment. And then once we've done that, we represent it as a matrix, and we can use linear algebra or stochastic simulation to learn more about those dynamics. So, for example, to see how long it takes for 90% of my innocents to be corrupted by the gossip. So continuous time Markov chain, as I say, you can think of it really as like a state machine with some extra information. It's a set of states, usually finite if we're going to use linear algebra, but some of the models I'll talk about are not. We have an initial state, and we have a way of labeling our states to know what's happening in them, and then we have a transition relation, which we typically represent as a matrix, and this gives us the rate of moving between the different states. And if that rate is 0, then there is no movement, so you can't reach the state S1 from state S2 if PS2 S1 is 0 in the matrix. Okay, so it's a very intuitive, easy model to build, and also has great benefits because of this memoryless property. So because we don't need to worry about how long things take, because in every state and at every point in time, it doesn't matter how we arrive there. Okay, so our future behavior only depends what state we're in, and it doesn't matter how long we've been in that state. Okay, so the exponential distribution is the only thing that gives us that property. So I'm going to talk about a particular stochastic process algebra called BioPepper, which, as you might guess by the name, was particularly developed to model biological processes, but it's more general than that. We use it quite often to model processes where we're interested in populations, and you'll see how that works in a moment. But the basic terminology that goes with BioPepper is very biologically sort of oriented. So we call the things in our system species because that's what they call them in biochemistry. So you can think of a species as being a protein or a DNA or something else. So it's not species as in cats and dogs, it's biochemical species. And our components, so the entities that we're interested in looking at their behavior, are given a quantitative state. So in other words, we just count how many of them we've got. So if we think back to the running example, I'm interested in how many innocents, how many repressors, and how many spreaders I have at any particular time. The same is true when I'm modeling biochemical reactions. I want to know how many of this protein, that protein, or molecules of water, say, that I have in my system. And so a component varies its state just by varying its amount, but we have to say how those are tied together. And those are the reactions in our system, and the reactions will vary the amount of a particular species by a number that's called stoichiometry. We're used to the idea of stoichiometry. We all know that water is H2O, so the stoichiometry of hydrogen in that reaction is 2. So this is the syntax. We have the idea of what we call sequential components. These are our species, the entities in our system. And we have an idea of prefix. It's a little bit more complicated than the one I showed you at the beginning, where it was just alpha.p. So now we have the idea of alpha and this kappa, and the kappa is the stoichiometry. So it's saying, when I do an alpha, how many things are involved? And then instead of just a dot, we have a whole set of different operators here. Because if you think about the reaction, say the innocents and the spreaders, for the innocents, when they meet a spreader and they spread, we reduce the number of innocents. So we say that's a reagent in that reaction. Whereas the spreaders are increased, so it's a product from that reaction. And so that's what these are. So this is a reagent, this is a product. And then these are, this is like an enzyme, so it increases the reaction. It's like an inhibitor. And then this is just something that needs to be present but is unaffected by the reaction. So these are just several forms of prefix, like we saw before. We have a notion of choice, so we may have two different reactions. So when we have, for example, a complexification in biochemistry, we can have A and B joined together to be AB, and then that may go on to have another reaction or may dissociate. So this is just usual choice, and then we have names for things. So these are how we build our basic components, and then we put them together to form a kind of soup that describes our system. And so these are, we put in parallel and say what they interact on, and each basic sequential component, we started off with a count. So we say how much of that we have to begin with. So we have this description that describes our soup, which will have the names of the species involved and their initial quantification. So you might say, well, there's no exponential here, there's no rates. We actually do that, oh, sorry, I forgot to. We do that separately. We say for each action type, we have a function that tells us what the rate should be. And then finally, we have to give this a semantics. We want to build a continuous time Markov chain. We actually build it in 2 steps. We have one step that's called a capability relation that just looks at what's possible, and then we build the stochastic parameters for that. So let's look at our example again. So this was how we represented it before, and this is how we represent it as BioPepper. So we should concentrate first on this, because this is the BioPepper description. So it's saying, if I have an innocent, the only action they can take is to be involved in a spread. That will decrease their number by 1. So whenever an innocent hears the gossip, they're no longer innocent anymore. The same reaction will be involving a spreader, and as a result, there'll be 1 more spreader, because the innocent now knows the gossip. And similarly for the other actions, so we can see our spreader can take part in the spread, or stop 1, or stop 2, and then repressors take part either as a product in both stop 1 and stop 2. So that tells us the capability, what these species can do. And then to understand their rates, we look at these kinetic laws, and it says that spread will have a basic rate of Ks, and it will depend on how many innocents and spreaders we have, because they randomly mix together. So we multiply those together, which is called mass action. For the stop 1, we need 2 spreaders to meet. For the stop 2, we need a spreader and a repressor to meet. And what I'm going to call parameters are these values Ks and Kr, and they tell us the actual basic rate at which those things happen. And in this model, and in all BioPepper models, those are given concrete values. Okay, so these are, in this case, 0.5 and 0.1. And then this is my description of the soup, if you like. In this case, I'm saying there are 10 innocents, 5 spreaders, no repressors to begin with, and the star just says, whenever there's a common name, you have to share it and do it together. And we can run this, we can run a simulation, and we would see the number of innocents decrease over time, the number of spreaders, depending. Here the repressor rate is quite low, so we would see a big spike in spreaders. If you increase the repressor rate, the repression will happen faster than people get to hear the gossip, and so you'll see a much lower peak in the number of spreaders. Okay, so this is just to give you the basic idea of a concrete model which has dynamics in terms of a continuous time Markov chain and a formal description here. So we have these 2 methods. So what I've just shown you is the mechanistic model, where we start from a description or a hypothesis and build this formal representation, and then algorithmically that will mimic the behavior of the system. Okay, we can run a simulation, for example, and we will step by step behave as if we were the system, and we can get data from that and then compare that data with the system we're modeling. The machine learning approach, we would instead start with the data and then try and refine our model based on that using statistical techniques. And if we compare them, well, one of the reasons that we do the mechanistic model or formal methods-based modeling is that we then build general execution engines. So we have the semantics of the language, we have a way of building the mathematics underneath it, and we can reuse that many times. So we only have to design the language, build the semantics, the mapping to a CTMC, and then we describe many different models using the same engine. The models can be used speculatively, so there might not be data, and we can just set up the model. We can play with the parameters to be slightly more unrealistic, perhaps, to see what would happen. And obviously, if you're based on data, you haven't got that opportunity. But the model does assume that I know everything about my system. So those parameters, KR and KS, I had to give them concrete values. But I might not actually really know, because that's just something that we would need to build from observations about the system. In contrast, the data-driven modeling has a much more rigorous way of handling parameter uncertainty. So it can cope with me just saying, I don't know what KS and KR are. I just have some idea of where they might be. But as yet, there's very limited treatment for stochasticity, so the kind of probabilistic systems that I'm interested in within data-driven modeling. And in many cases, what happens is that people have to build a bespoke system, so they build a special mathematical model for each example, and they have to do that every time. So you need a lot of expertise to be able to do the reasoning. So what we wanted to do was to bring these together. So we thought about, well, what about bringing something so that we can put uncertainty into our mechanistic models, and then automatically use observations to improve on that uncertainty while still doing it in a formal context so that we have a reusable engine rather than having to build a bespoke reasoning framework each time. So we took the basic process algebra biopaper and then looked at developing this new language which we call Proper, which is a probabilistic programming process algebra. So I'm going to tell you a little bit more about it. So you might have heard of probabilistic programming. It's gained quite a lot of popularity in the last few years. It's actually been around for more than 10 years with languages like IBAL and Church, but it's had a lot of resurgence or extra interest in recent years with languages like Fun, Anglican, WebPPL, Stan, for example, that you might have heard of. These tend to be modeling static systems. They're not looking at the dynamics the way that I am. But they're a good model for us because what they do is they build a high-level language. So it's this idea of having an engine. So you write something that looks like a normal computer program, but you put some uncertainty in there, and you have an interpretation that understands conditional probabilities. You can also access data or observations and then try and refine your understanding of the system based on that. So the typical workflow would be that you describe how the data is generated in something that looks like a programming language. But as I say, variables are left uncertain. You can specify certain observations, and these then are taken as constraints on what are acceptable executions of the program. And you can run the program forward then and generate data that's consistent with the observations. So what you would do is just stop a run that generates data that's not consistent with your observations. Or you can do what's called running the program backwards, which is look for the values of the uncertain variables that are going to match the observations that you have. So our objectives in designing Proper was to retain the features of a stochastic process algebra that we found useful for describing dynamic systems. So having a simple model description in terms of components, but then a rigorous semantics, which gives us an executable version of the model and also gives us this engine that's reusable in many cases, but incorporates some of these features from probabilistic programming languages so that we record uncertainty in our parameters. We have the ability to incorporate observations and then access inference mechanisms to update the uncertainty based on the observations. So this was our biopaper model of the rumor spreading, where everything is concrete. What we want to do instead to turn it into a proper model is we no longer give specific values for KS and well, it says KT, it should be KR. We just say we believe they're somewhere between 0 and 1. Okay. I'll point over here because I feel I've been neglecting you guys. We also put in some form of observations, and actually within Proper we have different forms of observations. I'm going to today just talk about the simplest, which is we can have a trace, so we can have a time series that tells us the value of the quantities of species over time. But we also have the idea of logical observation, so we can use a temporal logic to express that the probability of seeing this value within time 10 is 0.5 or something. So we have different ways of expressing our constraints or our data within the language. And then we also have a number of different inference approaches, the simplest of which is ABC, which approximate Bayesian computation, which is what I'm going to talk about today. But we do have a variety of other ones as well, which are much more sophisticated. So in terms of my program, now that it's a proper program or model, it doesn't look very different from the BioPepper, and that's a good thing because we had lots of tools already work with BioPepper. But now we have this idea of a distribution here, observations, and an inference technique. So previously, our BioPepper model was a continuous time Markov chain. We could map it to a representation in terms of states and rates of moving between states. But what does it mean now that we've changed, that we don't actually have rates, we just have some guess of a distribution that rates might follow? So a proper model cannot be interpreted as a continuous time Markov chain. It needs something else. It is actually a distribution over continuous time Markov chains. So if we have concrete values, as we did with BioPepper originally, then we get one continuous time Markov chain. If we were to have a set of values, k ranging from 0 to 5 in this case, we get a set of corresponding CTMCs as we instantiate it with each of those values. But what we've moved to is actually that each of our parameters now has a distribution, and so we get a distribution over CTMCs. Now, we may have multiple parameters, each with their own distribution, but we can still just build a distribution over CTMCs to represent that. And formally, in terms of mathematics, the structure is what's called a probabilistic constraint Markov chain, which looks very like a Markov chain in that we have states, initial states, and ways of labeling our states. But now we have here a constraint function rather than a rate function. And what our constraint function tells us is the set of acceptable values for rates for any transition in the system. And this is a generalization of a previous mathematical structure called constraint Markov chains, which worked for the discrete time case, and also just worked in the sense of saying this value is okay and this one is not, without giving you any idea of the likelihood or probability of those values. So we shift to having this constraint function now working with distributions. So now we build the semantics for proper, and we base it on the semantics of BioPepper. So just as before, we have the capability relation. You remember that part of our model description hasn't changed. We can still see what actions are possible for our different components within the system. But now our relation gives a distribution of rates for each transition. And we can build this using a formal system of semantics called the state-to-function transition system, FUTS, and then this gives us the underlying probabilistic constraint Markov chain. So when we think about these Markov chains, what are they? They are systems where you've got states. When you enter a state, you've got various actions you can take, and those actions, rather than having a rate, have a distribution over rates. So how do we interpret this? There are two different ways of doing it. The first is what we call uncertain Markov chains, where at the start of a run, if you think of simulating the system, you look at all these different distributions that you have for rates, and for each one you take a sample. Okay? And you pick one value for each of those parameters. And then you execute or simulate the Markov chain with that value for one trajectory. And then when you want to have another trajectory, you do another sampling and you get another trajectory. And this represents the case where we don't really know what the values are, and we can, with drawing samples of the possible Markov chains, and we can use the inference to try and decide which is the most likely. But these models actually have another interpretation, which we call the imprecise Markov chains, which is that there is something that you're not modeling, perhaps the temperature in which the system is operating, which means that the rate of the transitions within the system vary during an execution. So in that case, to build a trajectory, you start off, and each time you encounter a transition, you sample from the distribution. And then you get to the next one, you sample from the distribution. You may come back to the same action, but you would sample again and possibly get a different rate for that to reflect the uncertainty in the environment. So in our current work, and what I'm going to talk about today, we focus on the uncertain Markov chain case, so the first case where for any trajectory, we just fix the parameter values. We have some other work with imprecise Markov chains, and the interesting thing is that in the limit, the two actually amount to the same. So this is our system. We've got a proper model, which we can think of as like this. We've got parameters that have a distribution, and so we get a distribution of CTMCs. We present some form of observation, that might be a trace or a logical formula. We use an inference algorithm, and we get a refined notion of posterior distribution. And as I say, typically with the uncertain Markov chains, what we're hoping to do is narrow down to see which of the Markov chains gives the best account of what we're observing in the system. So we want to go back to Bayes' theorem. This was the one that told us how to interpret the posterior as the proportion to the prior and the likelihood. The difficulty is that we cannot do exact inference on the likelihood for the kind of dynamic systems that we're looking at. So we have to use approximate algorithms or approximations of the system. One of the things that we have within Proper is that it's a modular approach, so you can actually plug in different algorithms there. But I'm just going to talk about the simplest one. But this is the basic idea. We have our model. We compile it down to its mathematical representation, you use it with an inference algorithm and some results, to then infer. And then we can do the usual things we do with a model of building predictions or statistics or plotting outputs. So I'm going to talk about the simplest inference algorithm that we use in this dynamic context, which is called approximate Bayesian computation. And it's a very simple simulation-based approach. So it approximates the posterior distribution essentially just by running the model with samples of parameters and then seeing which satisfy our observations and which don't. So say these are our points. Our observations are a time series of particular values. This might be the number of repressors over time or spreaders over time or something here. And then we get a trajectory. We've picked some values for KR and KS. And in this case, we get a trajectory like this. We look at the distance between our trajectory and our data, our time series of points. And in this case, it seems quite large. And so we reject this. So this sample of these parameters, we say, doesn't actually generate any positive evidence towards building the posterior. But on the other hand, we pick different, we sample again, we run, get another trajectory, this one, then we can say this looks reasonably close and we accept this. And so we just do this repeatedly. Now remember, the same parameterization will give us different trajectories. So we do it many, many times to try and understand which parameters are more likely to give us answers that are consistent. Okay? So that's the very basic idea of approximate Bayesian computation. We use something slightly more sophisticated, but this is just to give you an idea. So we sample a parameter set from the prior distribution, simulate the system, compare the trace. If it's good, if it's within the accepted distance, we accept it, and we just put that as a mark in our posterior distribution. And if not, then we reject. And there are proofs, I don't know what happened there. There are proofs that this will converge to the true value with enough samples. So if we look at our example again, as I say, we're just going to use very simple ABC. we're testing this inference with time series data. What we did was we chose the values for KR and KS, and we ran a single trajectory to get some data to compare against. So it is only one simulation, and it gave us a set of points which we're then trying to compare against. And this is our prior distribution, because remember I said it was uniform between 0 and 1. And then this is what we get as the posterior. So the red lines show you the true values that generated the data we used as observations. And the blue crosses are sort of positive results when we ran a simulation with that parameterization. So we can see that actually for the parameter KR, it's quite tight. So here we get roughly all values between about 0.02 and 0.2. But for KS, for the spreading, it's much wider. Now this itself actually tells us something about the dynamics of the system, that this system is bimodal. And if you have KS actually lower than 0.2, then you get no spreading at all. Okay, so we get a much tighter value on here with the number for the repressors, and here we get this width, but you can see that it cuts off quite sharply here. And then it doesn't actually matter too much what your rate of repressing is as long as it's or spreading is as long as it's greater than 0.2. So that's a very simple example. There is a paper about this, and there's a lot more detailed biological examples where we look at various genetic switches. We've also done some modelling of mumps and the spread of mumps within the population in Scotland. But I want to talk about doing things the other way. So that was trying to bring the inference in some ways into the formal framework. And what I'm going to talk about now is taking ideas from the machine learning and using it to help us with formal models. So we're combining, again, mechanistic modeling and machine learning, but what we want to do is build a smaller model. So often, I've just shown you a very simple model, but often our state spaces, when we build these discrete state models, get into the millions, if not hundreds of millions. And even simulation becomes incredibly slow. And so we want to find ways in which we can abstract the models, make them smaller, but keep accurate results. And this is particularly true if we do what's called multi-level modeling. So this is often the case where we perhaps have different sort of levels within our model because we, for example, in this case, we think about individual agents who have behavior internal to themselves interacting in an environment which then influences how they interact with each other. So the simple case I'm going to talk about is actually chemotaxis in bacteria. So the bacteria have within them biochemistry that determines how they move in a nutrient field. And so the environment is influencing the behavior of the agents. We have another example, again, there's another paper where we actually look at slime mold, which is a two-way interaction, that the environment influences the slime mold in how they move, but the slime mold emits slime, which then influences the environment as well. But we're just going to do the simple case here where the agents are influenced by the environment. So we've probably all heard of chemotaxis, the idea that bacteria move towards where the nutrients are or away from a toxin, but they don't detect a gradient directly themselves. They're too small. Some entities are large enough that they can sample at their head and their tail and see a difference in the value or the concentration of a chemical. But E. coli that I'm going to talk about are too small for that, so we're going to see how they actually do that mechanism. So this is, as I say, looking at complex systems comprised of possibly many layers. We're just going to talk about two. And at each layer, we have a stochastic process that's looking at some kind of input and producing output. And the output of one layer will be the input to the next layer. So in terms of our bacteria, we have an internal layer that's looking at the biochemistry and how that makes the conformation of the bacterium. And then we have the higher level is how the bacterium moves in space. And some layers are very expensive or costly to simulate. And in that case, what we want to do is actually reduce them to a stochastic map or a function where instead of actually doing the mechanistic model, now we actually just literally use a function of input to output. So when we look at E. coli, this is a trace through a microscope of an E. coli in a nutrient field. And you can see that it kind of has two types of behavior. It has these long straight stretches, which are called runs, and then it has periods where it's called tumble, and it's reorientating itself. So it's basically turning on the spot. And it determines how to do this according to what it senses from its environment. And then this produces a biased random walk, which should move it towards higher concentrations. And these are complicated models for very simple mechanisms and take quite a long time. So I'm going to show you what's already not a full model, but a fairly realistic model, and it takes about 450 minutes to do a full simulation of E. coli in a field. So when we look inside the E. coli, it has a number of flagella or tails, and each one of those is coupled with what's called a motor, which is actually how it's anchored into the cell wall. And each of these flagella motor pairs have three different conformations, which are called semi-coiled, curly, and normal. Okay, and we'll see certainly normal and semi-coiled kind of makes sense when we see it. And the motor will rotate either clockwise or counterclockwise depending on the conformation. So we can see down here at the bottom when it's normal, it goes counterclockwise. When it's semi-coiled or curly, it goes clockwise. This is for each individual motor. And then we have rates here of moving between. So when we think of the cell, its states depends on the states of each of these flagella motor pairs, and there's typically between 8 and about 12 of these in each bacterium, and each of those can be in 3 different states. So we've already got quite a number of states. So this is a picture of our bacterium. It has these flagella motor pairs. So this is the hairy bit, and then this is what's called the motor. And then inside that motor, we have this mechanism, which is moving us between normal, semi-coiled, and curly, So we have that for each of those. This is what we call a receptor on the cell, and this is what we call a ligand. So this is the chemical that the bacterium is sensing from the environment. And then we have this M, which actually stands for methylated state, but you can think of it as memory, because what the methylation state tells us is what was the previous values of the ligand that I saw. Okay, so this is how it can detect a gradient, because it can't do it within its own body. It has to keep track of what it's seen previously. So these are the key parameters, L and M, that will determine how we react as a bacterium to these. And then when we're in the normal state, all the flagella will line up in this nice way, make a collective tail, and all rotate counterclockwise. And this will send the bacterium off on a run. If it's not like that, and they're doing counterclockwise, they all spread out, and this is why we get it turning on the spot. Okay, so that's how it works. And we express its state in terms of a very simple predicate, in this case, which is true if we have nobody in the semi-coiled state and at least 2 in the normal state. This is a very simple predicate, but this technique then works with more complex ones as well. So the transition rates for the FM from the one conformation state to another is determined by the concentrations in the chemotaxis pathway, which are captured by these parameters M and L. So we assume that these rates K minus, K plus, are a function of M and L, and actually this is just a kind of time delay, mu equals R. And these functions are highly nonlinear, and the ones that we use, so they're parameters of a Markov chain, but they're very complicated functions. And we take it from this paper by Sneddon et al. Of what those functions should be. So we can think of this now as a population, so it's slightly different to previously. Our species, in Biopepper terms, are actually the flagellar-motor pairs. Okay, so this is what we're looking at. And then their states that they go through are this semi-coiled, curly and normal. And we're counting how many of those that we have. And so we build a Markov chain where the states are counting these different values, so we call that a population Markov chain. Okay, just to remind you. And then if we want to simulate a trajectory of this system, what do we do? Well, we've got these two modes of or two levels of activity. First, we're looking at the movement part, which is represented in grey, and then we have the biochemistry part that's represented in sort of turquoisey colour. And so what happens is the bacterium will sense the current concentration, so it looks at where it is in the plate or field where it is, and see what the value of the nutrient is there in that particular concentration. That then feeds into the biochemistry representation of looking at that value of L and calculating a new value of M, and we can then look at what does that give us in terms of conformation, in terms of flagellars and motors, and then we evaluate, are we going to run or tumble, and then we let it run and tumble for our time step. So we choose what the delta T is, but that delta T will typically perhaps be a minute or something while it moves. They don't move very fast. And then we do the same thing again, because we can see that if it's run, it will have gone in a straight line from where it was, and we can see what the concentration is there, and so on. So that's our micro trajectories. It's mechanistic, it's describing exactly what's happening, but it's costly to do. This is the kind of output that we get. So this is a single trajectory. So they do follow a gradient, but in a rather random way. So here, blue is low concentration, yellow is high. So this is actually its starting point. It's sort of going towards the higher concentration, but they're not very determined E. coli. This is, if we look at a whole set of runs, 100 runs, all starting in the same place, we can see that they do make progress towards the higher concentration over time, but there's very wide variance in that. So what do we want to do? We want to reduce the complexity of having to look at each flagellar motor pair and calculate its Markov chain and then see what that tells us about the likely run and tumble and things. We just want to understand, if I'm in this kind of concentration, and I was previously in that one, am I going to run or tumble next? Okay. So we want to reduce it to a discrete time Markov chain. So in this case, we have two states, and we just have a probability of moving from one to the other or staying in the same state, and similarly here. So specifically, here what we're trying to do is replace the steps that we had in the previous simulation algorithm, where we were really executing the continuous time Markov chain to see what happened when we ran that, to then determine, do we run or tumble? We just want a decision process. Are we going to run or tumble next? And so we build this simple discrete time Markov chain where this is a run state. If I get a particular value, am I likely to move into tumble, or am I going to stay running? Okay, so these are our rates we're turning now into these probabilities, and these probabilities are going to depend on L and M. Okay, so really what it is Bernoulli values for each of the states, phi1 and phi2, and as I say, it depends on the current values that we've seen in methylation or the concentration. So we're reducing from having this whole mechanistic model to just having a lookup table, essentially, that we can say, okay, if I was running and now my value is L equals 6 and M equals 5, I should keep running. But if it's L equals 6 and M equals 2, then I'm going to tumble. That's the basic idea of what we're trying to do. It becomes much more efficient to run the model, but first we have to build that lookup table or map. So how we do that is using, again, Bayesian inference. And we assume that we're building our function in terms of Gaussian processes. So we assume that the way that we use the methylation and the concentration L is a smooth function. We run some small observations. Using our detailed model, we make a few samples of trajectories there and see what happens for particular values of M and L. Do we run or do we tumble? And then we use the inference to turn that into a smooth function so we can answer the question for every value of M and L. So technically, what we're doing is we're collecting a whole set of data points here by running our model a few times, and then we build this map which tells us the probability that we're running, so that phi is true for particular values of M and L. And essentially we're sort of building an inverse back from our samples to turn it into a cumulative probability function. Okay? And then once we've built that function, when we encounter a new value, say M star and L star, we can just use the value here, which is built on our function. So this is an example. These red points are the ones that we actually got from sampling, and then the blue is the imputed function. Now, in fact, we had some difficulty with doing this because what happens is that when you actually run the simulations with the E. coli, what they like to do is sit very close to this boundary. Okay, so this is the boundary between running and tumbling. So occasionally we throw them somewhere further away so that we get different values of L and M, and we can build the function. So we still just take a relatively small number of values and then build this function that we use instead. So now we can build our simulations. Instead of executing the inner step in terms of a full execution of the Markov chain, what we do is, as before, we sense the ligand contribution at our current place, we estimate the new value of M, and then we just sample this, we just do a Bernoulli trial. Am I going to run or am I going to tumble? According to the function that we built. And then we do that, and we move on, and then we sample again. So this is another trajectory, this time using the coarse grain process. And you can see again here, we started here, and it's moved towards the higher concentration. And you notice that this distribution, when we look at this 100 times, is very like the previous one. So we've lost very little information by throwing away the detail and just looking at the effect of the inner model. So before, what we were doing was we had this population continuous time Markov chain, which we fed into the decision of whether we ran or tumbled, which then moved us to a new position. We sampled, and then we ran this again. Now we just use this DTMC, which is essentially just this Bernoulli trial, and do the same thing. So the outer loop has stayed the same, but the inner loop has become far more efficient. So this was our training data. So this is where we actually ran our samples. But we then tried it in different fields. So that was a particular ligand field where we had actually what's called isotropic. So you've just got a concentration that's spreading from a certain point. So we have one that's non-isotropic, and we actually have one where we have a drip of nutrient coming in the center. So the field is changing all the time. And what we see here is at the top, the micro simulations with the full model, and below the abstracted ones, so much smaller. So in order to try and test whether our distributions that we're getting for behavior are really the same in some sense, we did a Kolmogorov-Smirnov test, and you can see that they're not exactly the same, but they're reasonably close. And if we look at it in terms of the table, these are the KS values, and then these are the p-values. And what they show is that the likelihood of getting this closer distribution by chance is very low in all these cases. And here we get the speed up. So in the original field, we got a speed up of about 8, and we got 9 in the non-isotrophic, and actually about 9 again with the dynamic field, which was not the one we trained in. So this gives us the benefit of being faster to simulate, and we keep the important behavior that we're interested in. So we want to see how these E. coli react in the field. Now, if we wanted to really know a lot of information about the flagellar-motor pairs, we've lost that because we've just reduced that to a Bernoulli trial. But for many cases, when we're doing this multi-level modeling, we want to understand the lower level mechanism, but actually once we've understood that and the role, how it interacts with the higher level, a function's fine. And so we can do this abstraction. So there is some loss of accuracy because the Gaussian process makes certain assumptions, but in this case at least it's highly accurate. And actually the one I talked about, the other one that's in the paper, where we have the slime mold and it's a two-way interaction, again we get good accuracy. We've shown in this case that we get consistency in different environments, so it's not the domain where we trained and found our function, but it still works well. There is a cost, so we have to build our function, so we have to have a certain number of training points, and this takes about 15 minutes to build. But this compares with 450 minutes of running time when we use the full model. Okay? And in our case, we build sort of synthetic data by running this abstracted model, but you could see doing this with real data that you perhaps collected. So just to conclude, mechanistic formal models and machine learning models can be used in very highly complementary ways. With proper, what we saw was we could bring uncertainty into the formal models and at the same time give a formal framework that can be used for parameter learning. And with the statistical abstraction, we can build our mechanistic and formal models and then use the machine learning to make them more easy to work with and to abstract. So thank you very much for your attention. I just want to thank my various collaborators on different parts of this work and the funders. It's all joint, well, apart from BioPepper, but most of the work is joint work with Guido Sanguinetti, who's one of my colleagues at Edinburgh, and then we've had two excellent joint students, Anastasis Georgias and Michalis Michaelides. And there are various papers that you can find on the different aspects of the work. So thank you very much.
We have time for questions, so I don't know whether you want to take questions yourself. It's possibly easier, but I will attempt to rush around and bring a microphone to people, or they can shout it if you like. Any questions?
Quick question. It seems that there is like fundamental assumption about the system when you start modeling it without prior knowledge. So, for example, when you try to model even the rumors or any epidemiology, you somehow tend to impose a structure on the states, right? So I'm just curious how knowledge somehow partially knows something about this thing. So, yeah, I think that we have some useful solution and then structure.
So we assume that we know the state space. There is a mechanism, so at the moment when we're looking for values, we don't allow the value 0. But if we allowed the value 0 for any of the rates, that switches to, but it doesn't add more. We haven't done any more work on that. There's a group in Stirling who've worked with BioPepper, and not with machine learning, but with genetic algorithms. And because BioPepper has this sort of very, you can have a species and then you add a reaction, or you can take away a reaction. It's quite easy to build recombinant forms, and they tried doing this. And again, they were looking at disease data rather than biological data. They had somewhat mixed results, but that's the only work I know. I mean, model finding, fitting, rather than parameter fitting is a much harder problem.
Thank you very much for a fascinating and broad talk. One part that corresponds to things I understand is the slide in which you tested the prediction of your models by making a statistical significance test relative to actual observation of E. coli. On that slide, it looks a lot larger than what I'm accustomed to teaching students. I wonder if you could just explain, possibly for other students of mine who think that P equals 0.5, that 0.05 is a good one.
Okay, so now you get into the bit that I understand less. But I should correct that we're not comparing real E. coli data with the model. We're comparing the two models. Because I must admit, on the train, I was looking at that and like, that's not what I thought. So I went back and looked at the paper, and that is what we've always said, that those are good p-values that allow us to reject the null hypothesis. I'm sorry, I'm not a statistician.
Well, I'll have to ask someone else about it in the future, I guess.
General question. Idea that we're always interested in explanation of what we're starting when we're doing machine learning. I mean, in the sense that what you're trying to do, or what you are doing, I guess, is you're starting off with an explanation, and then you're forgetting some of it. Then I'm just doing abstraction. Is that a reasonable way of thinking?
Well, I guess in some ways the explanation, we're building the explanation first in some ways. And yes, you're right, we kind of throw away the details of that, but that's still underpinning our function, because that's how we build our function, is by having that mechanism of understanding to begin with. So I think the explanation still remains true, so the E. coli still is doing its curly and coiled and what have you to determine whether it should run. But we're just saying we don't need to actually look at all those details. We can just learn how it makes the decision by looking at what input it has. So in the original model, we have the input, and then we have a whole lot of explanation about flagella and motors and things that tells us the output, yes, it runs, or no, it tumbles. And we just skip out that middle bit and just say, okay, this input will give you that output. But the explanation is still good. So in a way, this is a reason to do it our way, I guess, is build the mechanism first, and then get the faster model that you get with machine learning.
I mean, I'm fascinated by that, and would like to talk more, because it seems to me it relates to the sort of way that we're going with some language processing things, where we built some very detailed hand-coded models, and now we approximate them with machine learning methods, and maybe this is at least abstractly a way of thinking. Thank you. Other questions?
Thank you for your talk. Just wondering to what extent the models you present scale to a large number of stimuli transitions, and whether that makes some inference more intractable rather than
They scale bigger than the ones I've shown you today, because I've shown you our smallest examples because I wanted to give the two different strands of work. I think the biggest model that was used with the proper framework is probably the months, where we have probably about 10 different parameters. And that runs still reasonably fast, so we probably could push it further. With the statistical abstraction from the multi-scale, the slime mold has, I think, it has more predicates. So we just have the true false here. We get a bigger DPOC because we have, I think, 3 different predicates there. And so it becomes incredibly slow.
And I guess a natural follow-up is, just this framework, do you need to often have a large number of predicates and states in this model?
No, quite often not, because you're quite often looking, what you want to see in the next level up of the hierarchy of models is just, should I do this or shouldn't I do that? So it's quite often a decision. So actually having 2, 4, or say, it's quite often enough.
Other questions?
And I was wondering to what extent you can model a system that does have memory by simply encoding the memory in the state.
In fact, that's what I do with the second example. So you remember in the second example we had N, which was a Stuckman methylation, but you can also think of it as memory because the value of N is calculated on the previous value of L. So in fact, that is a memory, but it obviously enlarges your state space every time you do that. But yes, we can do that with CTMC.
Shall I shout?
Yeah, I mean, unless you wanted to do that in order to make me run over time.
By coincidence, we just had a 2-day workshop on probabilistic languages in the maths department here. And it's not my field as a specialist, but I know there was somebody there from the bugs team who was saying they're celebrating 30 years of bugs development. I was just wondering, I guess bugs was used more widely by biologists.
Yeah, and I don't know why, but it's not usually listed as a probabilistic programming language.
Yeah.
As I say, people tend to say church and IBOM were the first, but I am aware of bugs. But I'm not quite sure why it's not usually counted.
That's interesting. So the bugs team seems to still exist, and they're building a multi-processing version at the moment. And I wondered actually whether you think it might be more at the level of abstraction that you work at.
I've never looked at it in detail, I have to admit, but I should have looked. Thank you.
Okay, so I don't see any more questions. I have one final act before we move to the reception outside, which is that we always give people who have given the real election a certificate to say that you've actually been
I've done it. I'll put that in my office.
Thank you.
Thank you.
Final thing.